welcome to Chapter 27, the reproductive system. We're going to first talk about maleness and femaleness, and then a little bit about reproductive, and then Chapter 28 is going to continue. Okay, so the Chapter 16 was covered, our first chapter, and we covered endocrinology, which included the hypothalamic pituitary axis. And so there is a hypothalamic pituitary gonadal axis that we did talk about. GnRH, or gonadotropin releasing hormone from the hypothalamus, is released to the anterior pituitary. This causes the release of luteinizing hormone and follicle stimulating hormone to circulate in the cardiovascular system until it reaches a receptor. This would be in the gonads, and this is what's responsible for sexual maturation. Now, the gonads will release a sex hormone, or actually an inhibitory hormone called inhibin, which will then feed back and decrease your GnRH, LH, and FSH secretions. Okay, so let's first talk about cell division. And if we remember back to our cell division, which is actually the first class in this series, we remember the terms haploid and diploid, referring to half and the full complement of chromosomes. So here we have interphase, which is the period between cell divisions. And during this phase, the DNA is synthesized into two. So it goes really from, you can see one green line and one purple line to two lines. And there's a little button in the middle. This is called your centromere. And the centromere determines the number of chromosomes. So even though we do multiply our DNA times 2 during interphase of meiosis, the total chromosomes is still 2n. So we've got 46 chromosomes. The green ones, let's say, are from dad. The purple ones from mom. That'd be 23 each. Both have a complement of diploid chromosomes. And then after DNA synthesis, they're still diploid, and just that we have double chromosomes. You can see two chromatids with one centromere. And if we look down at this chromosome here, just to review, we've got the telomere, which is the ends of the chromosome, the centromere, which is the middle, and the chromatids, which are the arms. So this chromosome has two chromatids. It is still one chromosome. Now in meiosis, if we have already undergone interphase, our chromosomes are doubled. And the first part of meiosis is segregation of the homologous chromosomes. So the ones that you got from mom and dad, they're randomly segregated. And then the second part of meiosis, these sister chromatids are separated, and you get your haploid gametes. Now in meiosis 1, remember meiosis occurs in the gametes. Mitosis occurs in the somatic cells, all the rest of the cells that are not the sperm or the eggs. There's a very important event that occurs right after the interphase of meiosis 1, and during prophase, there is an event called crossing over. And what you have is you have homologous chromosomes. These are 23 from mom, 23 from dad, but each one is paired, so you can number them 1 through 23. Let's say this is number 1 chromosome, number 1 red, number 1 purple from mom. They have areas of similarity, which means that they're homologous, and therefore an event called crossing over can occur, and this is the exchange of genetic material among similar genes, where you get a mixture with mom and dad's genes. And if you can look, at the little tips, you can see that this chromosome, which is now in telophase, has a purple tip to it. So it has already undergone crossing over. This is a process in prophase 1 of meiosis between the homologous chromosomes. Now They subsequently line up in metaphase 1. The homologous chromosomes line up, and they're independently assorted. So it's random which chromosomes we get. We get number 1 from mom, number 5 from dad, whatever it might be. Then during anaphase 1, these homologous chromosomes get pulled apart to the poles, A for apart, and telophase is when two cells are formed. Now, since we have decreased the number of chromosomes by half, we are now at our haploid complement end of meiosis 1. And just to compare, let's review mitosis. On the left, we've got prophase, which is when the chromosomes condense. They go from chromatin to chromosomes. Nuclear spindle starts to form. The nuclear membrane breaks down. During metaphase, your chromosomes line up where the sister chromatids then segregate. So you see that even though you're getting division here, or a division of your chromosome, the complement of chromosomes in your parent cell, or in your parent cell and then your daughter cells, is still diploid. So you can see that we have segregated our chromatids, and each of the sister cells is still diploid, just like the parent. Now, if we go to meiosis, we're going to start with a similarly looking cell after interphase. These chromosomes have condensed and multiplied. And so we have prophase 1 of meiosis, where this tetrad forms. The tetra means 4. That just means the two homologous chromosomes that are paired up undergoing crossing over. These are both double chromosomes. And you can see during metaphase 1, the homologous chromosomes meet in the middle, and they independently assort at this metaphase plate. They then are segregated to each daughter cell, where each daughter cell is going to be haploid. Then you have basically a mitosis on top of this meiosis, where we have our double chromosome. This is in a haploid state. It only has 23. And each 23 goes to 23 because your double chromosome is getting split into your single chromatid per chromosome. So here we've undergone meiosis, 2 rounds, meiosis 1 and then meiosis 2, to yield 4 daughter cells that are, all 4 of them, are haploid and genetically distinct. And we can compare. So for mitosis, DNA replication occurs during interphase. That is the same during meiosis. The number of divisions during mitosis is 1, whereas during meiosis, the number of divisions is 2. Each mitosis has a PMAT. Meiosis also has a PMAT1 or a PMAT2. The synapsis of homologous chromosomes. This is another way of saying crossing over. It does not occur in mitosis. Crossing over occurs in prophase 1 of meiosis. The tetrads form, allows crossing over to occur among homologous chromosomes. Then the number of daughter cells that are produced is 2 for mitosis and 4 for meiosis. The roles of the cells in the body for mitosis, this is going to be all the somatic cells, tissue repair, renewal. And for meiosis, it's going to be the sex cells, the gametes, the sperm and the egg. Okay. So now we can look at some of the anatomy. Now that we know what's going on in the sex cells, here we've got a sagittal view of a male. In blue, we've got erectile tissue. And let me move my picture here. So when I show you a cross-section of the penis, you'll be able to see the corpus cavernosum. These form cavernosa, which are caverns that fill with blood. This is the main erectile tissue, the corpus cavernosum and the corpus spongiosum. So the Sperm is created in the testes have these seminiferous tubules in them, and that makes up the epididymis as well, or seminiferous tubules. Then there is a vas deferens or a ductus deferens, which is the tube that takes the sperm outside of the body. So first it stops at the ampulla of the ductus deferens, where it is further loaded with prostate fluid and seminal fluid from the seminal glands, as well as bulbourethral fluid from the bulbourethral gland. And so the prostate, this is present only in males, um, as well as the seminal gland. The bulbourethral gland is also found in females. This produces a lot of mucus for sexual intercourse. So we can go back and look at our vas deferens. That's the tube that is carrying the sperm out of the body. It then forms the ejaculatory duct and the urethra, which goes then out of the body. Urethra is your final duct. Okay, let's look at a little different view. So here's the penis again. We've got the testicles. Now the testicles have descended from the peritoneum during development. They've descended out of the peritoneum, but they formed a little pouch. It's called tunica vaginalis. And this is just a little pouch of peritoneum. This is the male anatomy. Even though it says vaginalis, this just refers to the little pouch. And so it's from the peritoneum. The testes have descended. So they are surrounded by a tunica vaginalis, but this has gotten cut off then from the peritoneum. And so it's just a little orphaned pouch. The layer of the testicles is the tunica albigine. There is then a spermatic cord, which carries nerves, blood vessels, and the vas deferens away from the testicles and towards the penis. We can see a skeletal muscle on the scrotum. This is called the cremaster. This is only present in males, and this allows some control over the testicles for somatic skeletal muscle. There's also a smooth muscle called the dartos, which is in the scrotum, and this muscle contracts and relaxes to generate heat for the testicles. Testicles need not quite body temperature, but a little cooler than body temperature. That's why they're located outside of the peritoneum. I do want to say one thing about this venous plexus. It's called a pampiniform venous plexus. This organ is highly vascularized. Okay, let's look at then a cross-section of a testis. We have the seminiferous tubules that make up the testes. We can see the tunica albigine making up the first layer and the tunica vaginalis, the surrounding layer of membrane from the peritoneum. And we have, if we follow the seminiferous tubules, a first round to the epididymis, or the head of the testicle, then the body of the epididymis, the tail, and then out the vas deferens. Okay, let's look at the bottom portion here, showing a cross-section of the penis. The dorsal is on top, the ventral is on the bottom. We can see a corpus spongiosum on the ventral side, and then two stalks of corpus cavernosa on either lateral side. And these, all of the blue, are erectile tissue, which fills up with blood and will cause the penis to erect. Now we can see the penis, and there's the prostate, and then the bladder. And we can look at this anatomy and appreciate how in some males the prostate will enlarge with age, and this has a tendency to push on the bladder to increase bathroom at night, having to urinate very often. It does take some room out of the bladder and add some pressure. We can see the urethra going out, the glands of the penis, which is the head of the penis. We have the body of the penis and the root. Okay, if we go inside histologically into a testis, we can see a number of different cells. We've got spermatogenic cells. These are your stem cells producing your sperm. So spermatogenic cells are simply the next level up of stem cell. And the spermatogenic cells are sustained by a second group of cells. These are conveniently called cystinocytes. So the cystinocytes will provide the spermatogenic cells with whatever they need and remove their waste. There's also interstitial endocrine cells. These are the ones that are going to secrete testosterone. So the interstitial endocrine cells will produce the testosterone that makes this person male. There are also some smooth muscle like cells, seminiferous tubules, and they can also contract and regulate temperature of these surrounding sperm. The sperm are the primary spermatids, actually, I should say, are in the middle of this testicle, and on the outside would be the stem cells. And as you progress towards the lumen, you are developing your sperm. Here's another look, but this histology, we're seeing the sperm tails. We can see they're organized. So we've got tails in the lumen of these mature sperm that are ready to be flushed out. Okay, starting with the spermatogonium, which is the stem cell, these operate by mitosis to produce subsequent cells, in this case a primary spermatocyte. As this enters meiosis, it will form 4 spermatids. And these are all haploid compared to the primary spermatocyte. And as the sperm develop, they lose their cytoplasm, and this bottom portion is a mature sperm. We've got the head, the neck region, the tail. The neck region has a lot of ATPs so that motility can be undertaken. Okay, another look at the testes. We've got the spermatogonium, or the stem cells for the spermatids. And as we progress towards the lumen, we prepare mature sperm that can be ejaculated. Here we can see some cystinocytes, which are sustaining our spermatogonium. So they help get rid of waste. They help give them components that are necessary. Okay, and so spermiogenesis starts with the circular cell, ends with the sperm type of cell, with the tail, the midpiece, and the head. We can see the cytoplasm has been lost, and with that, all the organelles that are normally present in the cytoplasm is lost. So the only thing that the male contributes to the egg is the nucleus of the sperm. The male does not contribute mitochondria, which has its own genome set, you might remember. Okay, let's then go back to our hypothalamic pituitary gonadal axis, where we've got gonadotropin releasing hormone from the hypothalamus. The anterior pituitary responds by producing follicle stimulating hormone and luteinizing hormone. These then enter the circulatory system, where they circulate until they receive their receptor, which is in the testes. The luteinizing hormone activates the interstitial cells to produce testosterone. The follicle stimulating hormone will activate androgen binding protein. Androgen binding protein is very important for making testosterone and making it available to these spermatogenic cells. One last factor, inhibin, is a factor that is released from the testes to the endocrine system, or back into the circulatory system, where it feeds back on the anterior pituitary to shut off the FSH and LH secretions. So inhibin will produce this negative feedback system where we only have FSH and LH in the absence of inhibin. There is a level of testosterone in the plasma that is maintained through the lifetime of the male. And so we have here months and years in age on your x-axis and plasma testosterone levels on your y-axis. The first portion would be in utero when we're still in your mom. We've got at fertilization, testosterone is going to peak at around 3-4 months, and then this baby continues to develop male until birth. There's another surge of testosterone, and then puberty is the next surge, and we see a dramatic increase in testosterone at puberty, and it's sustained through the lifetime of the adult male. Towards the end of the life, there is a decrease in plasma testosterone. This is a source of low T for adults, and some individuals will inject testosterone to get back to their high levels. So you can also overlay sperm production. This happens at puberty in the test in response to testosterone, and you can see the male produces testosterone or sperm for its entire life. Well, from puberty until death. Okay, let's move to the female. So we see this mid-sagittal section of the female. We can see the uterus. Okay, the uterus has a fundus. It's got cervix and two folds, which are called posterior and anterior fornix. The uterus is very thick. The myometrium constitutes the thick portion. The endometrium is the layer on the inside, which we'll talk about during menstruation. How about ligaments attaching the uterus? There are a number of ligaments that keep this uterus in place. In posterior, there's a uterosacral ligament. The anterior is the round ligament. Then there is a suspensatory ovarian ligament. And we just are missing one more ligament, the actual ovarian ligament, that is not in this picture. Okay, from this mid-sagittal section, we can see the rectum connected to the cervix and the urethra. Now you can see the urethra connected to the bladder is not part of the reproductive system. So with the vagina, that's the reproductive part, but the urethra is not. That's a common question, which will be in exams, and it's compared the male versus female. The male, the urethra, is part of the reproductive system and the excretory system, whereas the females, the urethra, is just part of the excretory system, not part of the reproductive system. Now, the female also has erectile tissue. This forms the clitoris. And you can look at some of the anatomy of the uterine tubes. Uterine tubes conclude with the infundibulum and fimbria. These fimbria kind of collect the egg that has ovulated from the ovary, and there are little hairs on the uterine tube which will guide this egg down into the uterus for implantation. Okay, let's look at the ovary. Here we can see the ovarian spensatory ligament, as well as what looks like the round ligament of the uterus. If we look at the ovary, we can see a number of follicles on the inside, and these follicles have varying development. There's usually a primary follicle, which is the one that's active at the time, and these will alternate between left and right ovaries every month, which follicle is the primary follicle. So we see the layers of the ovary. We've got the tunica albigine, just like in the testi, and then follicles. Okay, let's look at these ligaments. So posterior of the uterus is held by the round ligament. You've got the ovarian ligament holding in the ovary. fundus is the kind of the head of the well, the head of the uterus, and then we've got the body, and then the cervix would be the exit into the vagina. So we can see the fallopian tube connecting to the uterus, and in this tube there is an ampulla where the egg is going to travel until it gets to the uterus wall. Now, the uterus itself is made up of an endometrium, the interior lining, then a myometrium, the major muscular part of the uterus. This has to accommodate the baby for 9 months, so it stretches quite a bit. You can see it's at rest. It's quite thick and muscular, and then a perimetrium on the outside. Also holding the ovaries is a broad ligament, and this is not as strong as the ovarian ligaments, but this is a sheet of ligaments that help to keep these parts in place. Okay, the endometrium is highly vascularized, and once puberty happens, there's menstruation every 28 days. And during this time, the endometrium is lost. All these blood vessels are lost. And then there's another phase where they reproliferate, where the endometrium is built back up. These blood vessels are built back up, and the cycle starts again. There are spiral arteries. These are part of the cycle of regeneration, degeneration. Okay, so we talked about spermatogenesis, and that was with the sperm developing. We went through meiosis, and we produced 4 haploid spermatids. In the female, the process of oogenesis, and we can see on the left is the follicle development phase, and on the right are the meiotic events. So they're just side by side. We can see the primordial follicle is the initial follicle. This is the primary, what contains the primary oocyte. So the primary follicle contains the primary oocyte. And when the female is born, all of the eggs are in the primordial stage. And so a female is born with all of her eggs. They start meiosis, but then they have rest in prophase 1. And so this is the crossing over phase. This is just before birth. So by the time the female is born, she has all of her eggs for her lifetime. And then at puberty, the cycle starts where each egg is dropped into the uterus and flushed out if not fertilized. So the process of oogenesis is a little different than spermatogenesis in that you don't get 4 eggs as a result of meiosis 2. You only get 1 ovum or 1 oocyte and 3 polar bodies. Okay, these are the nuclei which contain the chromatids. These are all haploid. Are the organelles, the growth factors, the cytoplasm. That's all really important for the sperm and the egg to develop the zygote. So that's why meiosis for the females produces 1 ovum and 3 polar bodies which are just reabsorbed. These polar bodies are not used in fertilization, and they are not stored in uh, the follicles. So if we look at male versus female side by side, the total time to produce 1 gamete, males is about 74 days. That's just how long it takes to go from a spermatogenic cell into the primary spermatid. For the female, it takes anywhere between 13 and 50 years for to get the product of that meiosis, and 50 years would be about when menopause starts. So the occurrence during the lifetime, the males produce sperm from puberty to old age, depending on their levels of testosterone. The females will begin oogenesis at birth and then end oogenesis at menopause. The number of gametes per division for the male is four spermatids per spermatozoon, really. The females, just one large ovum is produced per meiotic division. The number of gametes per lifetime, the male is more than 1 trillion. The female will be less than 500. Every female has a different number of eggs that they are born with. Error rate. This talks about chromosome segregation during meiosis. Sometimes these chromosomes get sticky when they've been arrested for a number of years. So the error rate actually increases with the age of the mother or the father. For the parent, for the father, the error rate is about 5%. For the mother, the error rate is about 20%. And that just means that 20% of the time that the baby has the wrong chromosomes, it's from the mom's meiosis, and 5% would be from the dad's meiosis. What about the cells surrounding the gametes? Well, male's spermatid has one cystinocyte that will sustain many spermatids. For the female, there are granulosa cells, and there are many granulosa cells that sustain one ovum or one oocyte. So for the males, one cystinocyte will maintain several spermatocytes. The female, one oocyte will be maintained by several granulosa cells. Okay, we have side by side showing meiosis, results of the ovary itself and the follicle, and then the ovum. So we see that before birth, the cells in the female will arrest in prophase 1. Then at each cycle, there is a primordial follicle, which then develops to a primary follicle. This develops to a secondary follicle, into the vesicular follicle. Then we have ovulation, the point at which the egg leaves the follicle. And then the follicle turns into what's called the corpus luteum. This is a ruptured follicle, but it still will give off a lot of hormones. So if we look at the cellular events going on with these follicles, we've got before birth, the oocytes, or the oogonium, arrested in prophase 1. Each cycle after puberty, a primary oocyte is developed, and this develops an ovum, one ovum, through meiosis 1 and 2. Also develops 3 polar bodies, which are reabsorbed. Okay, let's look more at the ovary. The ovary has a number of follicles in it at different stages. And so there's a primordial follicle. This is the primitive follicle. Develops into a primary follicle, into a secondary. And then this follicle will enter a vesicular phase and a luteal phase. So we see the follicle is developing, it's now mature, and you've got ovulation, which is release of the oocyte from the follicle. Now the follicle is called the corpus luteum, and it will continue to be active even though it has ejected the egg. So if we look at the different phases, we've got follicular phase and luteal phase. I'll show you in another picture that is a little bit easier to understand, but let's look at the hormones that are responding. Let's start with gonadotropin releasing hormone from the hypothalamus, leading to FSH and LH from the anterior pituitary. FSH is then going to act on this follicle and activate androgen production from granulosa cells. There's also inhibin that is produced, which will feed back on the anterior pituitary and the hypothalamus to shut off the signaling. This is why it's a negative feedback mechanism. So this was follicle stimulating hormone acting on the follicle, activating granulosa cells to produce different type of androgens to estrogens. Estrogen is what's important, and there's a particular enzyme that takes the androgen or testosterone and produces estrogen. The enzyme responsible for that is called aromatase, and this happens in the thecal cells in response to luteinizing hormone acts on the thecal cells to release androgens, which are then converted to estrogens by aromatase. This is called the follicular phase, where the follicle is the main area of interest. Then there is an LH surge. Okay, what happens is the mature follicle starts to produce a lot of estrogen, and this estrogen will eventually feed back on the hypothalamic hypophyseal gonadal axis to release an LH surge, or a surge of luteinizing hormone. This is going to cause ovulation. This is the midpoint of the cycle, and the egg has been released. This is now the secondary oocyte, and the follicle then, in absence of the oocyte, develops into what's called the corpus luteum. And the corpus luteum continues to pump out hormones. And this time we get high progesterone, high estrogen, and high inhibin. Now the inhibin is going to inhibit the LH surge, whereas the progesterone and the estrogens are going to continue to inhibit the LH surge. I'll show you a picture of that. So here we have the cycle, the follicular cycle. We can see different phases, which we'll go into, but in the middle of this cycle, between the blue and the green, is ovulation. So luteinizing hormone is surging, and at the top, then we've got ovulation. We've got this is released. Similarly, follicle stimulating hormone is released. We can now make out a follicular phase and a luteal phase. The luteal phase is due to the corpus luteum. It has degenerated, and now it continues to pump out a lot of hormones. We've got ovulation in the middle, and the follicular phase, or development of the follicle, in the beginning. So the follicular phase includes the dominant follicle. We've got LH surge causing ovulation. We've got luteum producing estrogens, progesterone, and inhibin. The inhibin will shut off the LH surge, where the estrogens will promote development of this egg. Now, if we look at estrogens versus progesterone, there's a spike in estrogens just around the LH peak, but then the levels of progesterone increase over this phase, and this is actually the function of some birth control, is to clamp the female at high levels of progesterone. Progesterone inhibits the release of FSH and LH, so ovulation never occurs. So if your progesterone levels are high and they're clamped at a high level, ovulation will just not occur. And then there's usually taking birth control, a water pill option that will then allow progesterone levels to decrease where ovulation can occur. So this is the basis of some birth control, clamping progesterone at high levels. Okay, let's look at the uterus. We've got uterine phases, proliferative phase, which is development. The menstrual phase actually starts with menstrual flow, and this is the first 4 or so days, where you have menstrual flow and the endometrium has been degraded down to its lowest point, the nadir, or the lowest point in this graph. The beginning of the proliferative phase. Then we have proliferation of the endometrium, lot of vasculogenesis and angiogenesis, where you have a lot of blood vessels that are formed. Ovulation occurs at the end of the proliferative phase, and then the secretory phase starts. Hormones that are released from the ovary. And if we take these 5 or 4 graphs and put them together, we can see that the proliferative phase is maintained, and then high levels of estrogen lead to high LH, ovulation, and now we've gone into our luteal phase, where our further ovulation is shut off by progesterone. And this cycle then will end in the secretory phase. You'll have menstrual bleeding. I think that's the end of the cycle and the beginning of the next cycle. Okay, let's just summarize the hormonal effects of the estrogens, progesterone, and testosterone. So the estrogens, the major supply is the ovary. These are mostly estradiol, and that's created through aromatase action on testosterone. The stimulus for estrogen's release is the high follicle stimulating hormone and luteinizing hormone. They both negatively feedback on gonadotropin releasing hormone, so it's a negative feedback mechanism. The estrogens are the secondary sexual characteristics of the female and stimulate genesis and ovulation. So let's look at progesterone. Progesterone is mainly from the ovary, but from the corpus luteum. Progesterone is released after the LH peak, and then progesterone starts to be produced by the corpus luteum. Testosterone is produced in the testes, in the interstitial endocrine cells. LH is what causes testosterone to be produced. And we looked at androgen binding protein, which keeps the concentration of testosterone high and keeps the testosterone near the spermatogenic cells. Okay, and lastly, let's look at sex chromosomes. We talked about 46 chromosomes per person, 23 from mom, 23 from dad. Well, the first 22 are somatic, and then the last one is the sex one. So our sex chromosomes determine our sex. Everybody gets an X. 2 X's is a female, and X and a Y is a male. So presence of the Y chromosome is what creates maleness, you can say. And you can have extra chromosomes. We see that in a number of different conditions. Okay, that concludes our chapter. I'll see you next for chapter 28.